Hi, my name is Andrew Kershaw, um, and this is my Diseases of the Cell project on hemolytic anemia. Okay, so what is hemolytic anemia? Hemolytic anemia is a blood disorder that can be globally categorized as counts of red blood cells that are below the normal reference ranges of 4.0 to 5.6 million cells per microliters in males and 3.9 to 5.1 million cells per microliters in females. Now, hemolytic anemia is interesting in that it's this broad term. It's actually more of a symptom of a large number of potential causes of anemia, and therefore can be classified in a number of different subtypes. This makes it a great area for research and for active discovery today. So, to explain hemolytic anemia at the cellular basis, let's start with the life cycle of a red blood cell. First, red blood cells are produced and released as immature reticulocytes in the bloodstream. From there, the reticulocytes mature into red blood cells. The red blood cells will operate to deliver oxygen to your tissues and remove CO2 from your body over the course of 120 days. After 120 days, roughly, the red blood cell will be broken down. It will be broken down into heme, iron atoms, globin, free hemoglobin, and lactate dehydrogenase, among other things, of course. And these materials are either recycled or excreted from the body. And in a normal individual, this process will continue for life. However, in an individual with hemolytic anemia, there will be an abnormal increase in red blood cell destruction, which is problematic. Therefore, there will be an abnormal increase in serum concentrations of red blood cell and hemoglobin materials. This increased concentration can be used in initial diagnostic testing for anemia, where you will find increased concentrations of lactate dehydrogenase, bilirubin, among other things. Therefore, at the organ system level and at the metabolic level, if we think more broadly about what's happening, hemoglobin is being destroyed. Therefore, what we know about hemoglobin, less oxygen is being delivered to the body and less CO2 is being disposed from the body. In the absence of oxygen, also known as hypoxia, cellular respiration, meaning the cell's ability to produce ATP, is reduced. This is due to the oxygen's ability basically role as a main electron acceptor at the end of the electron transport chain in the cell mitochondria. This results in a reduction in the cell's ability to maintain cell volume, trigger required biochemical pathways, and facilitate cellular communication in specifically the case of neurons. This means a person with hemolytic anemia can really expect increased baseline fatigue, right? As well as, let's go back, baseline fatigue and brain fog, given they're at a relative disadvantage when it comes to ATP production in the body. Additionally, increased levels of iron and free hemoglobin processing can lead to decreased kidney function over time. Further, all organs in the basically reticuloendothelial system, so think liver and spleen, can end up being enlarged due to like an over hyperproductivity of constantly reducing and getting rid of red blood cells. So, shifting to etiology, hemolytic anemia can be categorized into two broad buckets. The first is intravascular, which occurs when red blood cells are destroyed in the blood vessel itself, and then extravascular, which occurs in the liver and the spleen within the reticuloendothelial system. Within the intravascular bucket, there are induced and autoimmune forms of anemia. So the main induced forms of anemia are mechanical valve, acute transfusion, osmotic-related hemolytic anemia, as well as microangiopathic hemolytic anemia. Autoimmune forms of anemia include cold immune hemolytic anemia. This is when antibodies recognize and attack antigens on red blood cells at temperatures that are below the normal body temperature. Antibodies bind to the receptor on the cell membrane and then bring them to be destroyed by phagocytosis. This is due to a mutation in the gene KMT2D and CARD11. it is not known to be inheritable as well. Now, paroxysmal nocturnal hemoglobinuria is a form of anemia. This is when red blood cells are actually destroyed through complement activation. And this is due to a mutation in the PIGA gene, and it is inheritable via autosomal dominance pattern. Warm immune hemolytic anemia is basically the same mechanism as cold immune hemolytic anemia. However, it is triggered when temperatures rise above normal body temperature. This is related to a genetic mutation that's still being researched, and is not thought to be inheritable as well. Within the extravascular bucket, we have different types of anemia, including hypersplenism, infection of red blood cells, inheritable forms of extravascular anemia, as well as hemoglobinopathies. This is when there is basically a defect in the way your hemoglobin functions or is basically put together. An example of this is sickle cell. This is due to a mutation in the HBA1 and 2 genes, and this is a disorder that is autosomal dominant in inheritance pattern. Another form is cell membrane defects. This is when the way the red blood cell's membrane is composed is defective. This is like different morphologies that actually cause the red blood cells to get stuck in different capillaries and ultimately be destroyed. This is due to a mutation in the ANK1 gene. This is a disorder that is autosomal dominant. Finally, we have different enzymatic defects within the red blood cell. So one is the G6P defect, and this is when in red blood cells, anaerobic glycolysis is the main pathway to produce energy in the form of ATP. During this process, there's free radicals that are created. G6P is an enzyme which helps reduce free radical formation. When G6P is deficient, free radicals build up and ultimately lead to the destruction of the red blood cell. This is due to a mutation in the G6P gene and is thought to be X-linked recessive in terms of inheritance pattern. One last note, you'll see this dotted line, is that warm and cold immune hemolytic anemia, according to the research articles I read, could actually be described either as intravascular or extravascular. I group them as intravascular for the sake of this diagram. However, just know you can also consider them extravascular. Also, hemolytic anemias are often inherited and therefore represent a mistake at the gene level. So they're not, rather than like some issue occurring during a cell cycle. So patients suffering from hemolytic anemias usually like a normally dividing cell. However, the cell is deficient in other ways. So that's important to know. All right, so we talked a little bit before about diagnostic approach for hemolytic anemia, but I thought I'd touch on it one more time using this workflow to illustrate how the clinician and care team staff can work together to figure out which anemia is impacting their patient. The first test typically used is a hemolysis workup, and this measures for constituent material of the red blood cell that we discussed before. So think lactate dehydrogenase or bilirubin. So that hemolysis workup is trying to measure those concentrations. If they're elevated, that's a problem, and it typically indicates anemia. So that's our first test. Okay, so the next test is a peripheral blood smear. So, okay, we've established they have anemia. Let's figure out what type. A blood smear is a test that is performed usually by a pathologist that will be used to diagnose a type of anemia if possible. And they'll diagnose it based on morphology, but today also they use forms of genetic testing to figure this out as well. Lastly, if they can't figure it out, from these two tests, they will move on to a direct antiglobin test if they're thinking, this might be something that's autoimmune related. And so this will be used to identify those autoimmune anemias that we discussed. So important to note, these tests don't come to mind usually during a routine visit to the doctor. There's something that symptoms would dictate the use for. Further, if you're found to be anemic, you may be taken care of by your primary care doctor or referred to a hematologist for specialty care. All right, so treatment strategies. I also wanted to touch on different treatment strategies the medical community uses to manage hemolytic anemias. We'll just touch on a few of these. I'm bumping up against time, but for things like induced anemias that we talked on before, often the treatment strategy is just to stop what's causing the anemia, or to heal from an infection, or to manage some other issue or disorder that's causing your anemia. And so those treatment strategies are typically pretty straightforward and established, and don't lend themselves to much experimental testing. So one is acute transfusion reaction. The prevention is just to obviously not be transfused with a blood type that doesn't match your blood type. And then standard treatment would be to stop that transfusion and to reestablish the patient's blood type and transfuse them with the correct one. However, in like forms of autoimmune hemolytic anemia, the treatment strategies are a little bit more complex. So prevention is hard to do in this case, since it's likely maybe genetic, or something that you uniquely have. So you just need to plan for it and plan around it. But standard treatments include steroids, obviously staying out of the cold and warm, immunosuppressants, things like that. Experimental treatments are different B-cell therapies, phagocytosis inhibition, plasma-directed cell therapies, as well as complement inhibition. Things like hemoglobinopathies, genetic counseling is effective. Treatment strategies will vary by the different type, but typically gene therapy is something that is used now to help manage the effects of this disorder. So, very similar with cell membrane defects, so genetic counseling to diagnose this and to maybe prevent from potentially passing this on is important, but standard treatment strategies include some forms of medication. However, the common treatment strategy is also splenectomy, which is a little bit invasive. Enzymatic defects, so that G6P disorder that we talked about, prevention, and this is actually an epigenetic component that's was kind of unique to this one, is avoiding naphthalene, which is kind of a rare chemical anyway, but it is something that can promote the expression of this disorder at the gene level, or at least studies have shown that. And so this is certainly an epigenetic factor. You want to avoid this if you're at risk for having this disorder. Standard treatment, though, for something like this would be a splenectomy. Okay, so now that we've touched on treatment strategies, let's just finish out with research trends. So when searching hemolytic anemia on PubMed, the article count has been rising over the last 100 years, topping out at 2,974 articles, new research papers published in 2021. So I would say the outlook on the disease and interest in research is long-term and bright and continues to grow. New advances are shifting the treatment options away from splenectomies, which are a little invasive, and moving towards more of like a secular drug regime, similar to the nature of the drugs given in cancer treatment. One is rubiximod, which is basically facilitates receptor-mediated cell death of abnormal red blood cells. So abnormal cells, like an abnormal sickle cell or something like that. Further, there are new drugs being created and studied for the treatment of cold and warm autoimmune anemias as well. One such drug is sudamimab. I'm terrible at pronouncing these drugs, but this one targets and binds to a protein called C1, resulting in complement inhibition, which is an active protein that binds to abnormal red blood cells to promote destruction. And this is big because this marks a shift in the treatment of these autoimmune hemolytic anemias that is more focused on adapting the body's native immune system to coexist with these abnormal red blood cells versus treatment options that were focused on destroying them. And then finally, I think the biggest area of new research and new discovery here is gene therapy. So there were many articles focused on using gene therapy to target these cells and even downregulate the expression of the creation of these defective genes. So this was big. So one of the biggest articles I read was an experiment that actually was showing capabilities of actually like reversing sickle cell disease. That was a durable effect for multiple years using a gene therapy technique. Further, this gene therapy is being used to direct the development of drugs that we talked about, these new experimental drugs for autoimmune hemolytic anemias. Some include, these B cell directed therapies, complement inhibition, and plasma cell directed therapies as well. Great. And so that was my presentation on hemolytic anemia for the Diseases of the Cell project. Thank you very much.